Imagine treating a really deadly cancer, right? You watch the blast cells completely vanish from the blood smears, and you see the scans come back totally clear.
Right, you think you've won. You think the nightmare is over.
Exactly. But then maybe a year later, the disease just roars back, and it's far more aggressive than before.
Yeah, it's devastating.
It really is. And you realize the initial treatment didn't actually kill the cancer. It just, well, forced it into hiding. It basically triggered this survival mechanism that essentially trained the disease to become invincible.
Which is unfortunately a very common reality in oncology.
Yeah. So, welcome to this custom-tailored deep dive. For you listening, our mission today is decoding a really fascinating, highly detailed scientific research paper, specifically paper S4139202602697 We are going to explore how scientists have uncovered exactly how this hiding mechanism works in a notoriously difficult blood cancer, acute myeloid leukemia, or AML.
And, we are looking at a pretty dense piece of research here. It is packed with cellular pathways, genetic mutations.
Stuffy stuff.
It is. Lots of intricate survival strategies, but the goal here is to extract the vital mechanisms for you. We want to look past all the raw data and understand this specific protein that acts as this cloak of invincibility for leukemia cells. And more importantly, how researchers have finally figured out a way to rip that cloak right off.
I love that. So let's just lay out the clinical reality driving this research because it is incredibly stark. The statistics in this paper show that AML relapse occurs in up to 40% of younger adults.
Wow.
Yeah. And a staggering 80% of older adults.
It's a massive hurdle.
Right. And the standard treatment, which is these intensive chemotherapy protocols relying on cedarabine and an anthracycline, it frequently fails in the long term. It clears the bulk of the disease, sure, but the cancer almost always finds a way to return.
So to understand that really high relapse rate, you have to look at the cellular hierarchy of the tumor itself.
Yeah.
Because, leukemia isn't just this uniform army of identical, rapidly dividing cells. It actually contains a very rare, highly specialized subpopulation known as leukemia stem cells, or LSCs. These are essentially the true architects of the relapse.
So treating AML is kind of like weeding a garden by only ripping off the leaves, right? The bulk of the cancer dies, but the roots...
LSCs.
Exactly. The roots are left behind in the soil, which in this case is the bone marrow, to just grow back later.
If we connect this to the bigger picture, the most logical next step would seem to be designing a targeted therapy that just seeks out and destroys those specific LSC roots. But historically, we run into a massive targeting dilemma.
Because they look too much like normal cells.
Exactly. The biological markers typically found on the surface of leukemia stem cells, these are proteins like CD33 or CD123, they're not unique to the cancer. They're also present on normal, completely healthy blood stem cells.
And those healthy stem cells are the genesis of your entire blood and immune system. So if a therapy is designed to nuke anything carrying a CD33 marker, the friendly fire would be catastrophic.
Yeah.
You might eradicate the leukemia roots, but you'd simultaneously wipe out the patient's healthy bone marrow?
Leaving them with absolutely no ability to produce white blood cells, red blood cells, or platelets. It's a total biological minefield.
Which is why finding a new target is so crucial. So how did they do it?
Well, researchers have spent years searching for a therapeutic window, a marker unique to the LSCs that leaves healthy stem cells completely untouched. And since friendly fire makes traditional markers useless, they had to look outside the standard leukemia playbook.
And that search led them to this cell surface protein called CD81.
Yes, CD81.
Now, CD81 is a type of protein known as a tetraspanin, and previously it was mostly known for its role in B-cell lymphomas. But this paper really maps out its massive hidden role in AML.
Yeah, the researchers started by looking at a huge cohort of patient data just to see if CD81 was even relevant in AML. They analyzed 252 diagnostic samples.
That's a lot.
It is. And they looked at 38 relapse samples, too. And the expression pattern they found was highly polarized. They observed this bimodal distribution.
Meaning patients rarely sat in the middle of the spectrum.
Exactly. They either had leukemia cells with barely any CD81 on the surface, or their cells were completely coated in it.
So what does this all mean?
Well, finding yourself in that high CD81 category is a massive danger signal. The clinical data showed that high surface expression of CD81 at the time of diagnosis strongly correlates with significantly worse relapse-free survival and worse overall survival.
Oh, wow.
Yeah. And the researchers noticed this was particularly pronounced in specific genetic subsets of the disease, specifically non-core binding factor, AML, or non-CBF AML and NPM1 mutant AML.
You know, that NPM1 mutation detail is particularly interesting because the NPM1 gene normally codes for a protein that shuttles other molecules back and forth across the nucleus.
Right.
So when it mutates, cellular transport just goes haywire. So seeing this massive buildup of CD81 on the surface of cells in these specific patients, it kind of suggests the leukemia is actively exploiting this transport chaos to armor itself.
And we actually see direct evidence of that armoring process when we look at the paired patient samples. The researchers examined the exact same patients at the moment of their initial diagnosis and then again when their cancer relapsed.
Let me guess, the CD81 levels went up.
The jump was chilling. Surface expression leaped from 44% at diagnosis to a massive 71% at relapse.
Wow.
The disease was actively selecting for and enriching itself with this specific protein as it weathered the onslaught of chemotherapy.
Wait, I have to push back on this a little bit.
Okay, go for it.
Wait, so is CD81 just a marker, like a check engine light on a dashboard indicating the cancer is bad, or is it actually the engine driving the cancer off the cliff?
That's the perfect question. And the researchers knew they had to prove causation, not just correlation. So they transitioned from analyzing patient blood samples to manipulating the genetics of laboratory cell lines.
How did they do that?
Well, they used genetic engineering tools. They forced cells to overproduce CD81. Specifically, they overexpressed it in U937 cells. And then in other models, they used short hairpin RNA, or shRNA, to silence the gene entirely in OCI-AML3 and HNT34 cells, basically shutting down CD81 production.
And the behavioral shift in those modified cells was night and day, right? Pumping up the CD81 levels gave the leukemia cells immediate, measurable superpowers.
Superpowers is a great word for it. They became highly resistant to standard chemo.
Daunorubicin and Cerabine.
Exactly. And when they injected these cells into immunodeficient mice, these high CD81 cells didn't just survive, they thrived. They engrafted much faster, they created a vastly higher tumor burden, and they aggressively infiltrated bone marrow, spleens, and even adjacent muscle tissue.
Okay, let's unpack this. How is a single protein doing all that?
We have to look at the physical mechanism. A protein just sitting passively on a cell membrane doesn't magically repel toxic chemicals. The researchers discovered that CD81 physically alters the architecture of the leukemia cell. It induces the formation of these Filopodia-like membrane protrusions.
Which we can essentially compare to microscopic Velcro hooks.
Exactly.
These aren't smooth, floating cells anymore. They are bristling with these hooks, allowing the leukemia to aggressively latch onto its environment. Specifically, they bind to a protein called fibronectin.
Yes.
And they adhere really tightly to bone marrow fibroblasts, like the HS5 and MS5 cells, which are kind of the structural scaffolding cells inside the marrow cavity.
And here's a fascinating quirk from the paper. Once those leukemia cells use their CD81 Velcro to lock onto the bone marrow stroma, a biological trigger is pulled. The physical act of adhesion induces something called a G2 cell cycle arrest.
Meaning they pause their own division.
Right. They essentially hit the pause button on their own replication.
And pausing division might sound like a victory to someone, but it is actually a brilliant evasion tactic.
Oh, absolutely.
Because standard chemotherapy is specifically designed to destroy rapidly dividing cells, right? It targets the phases of the cell cycle where DNA is actively being synthesized or where the cell is physically splitting.
Exactly.
So if a leukemia cell uses its Velcro to anchor into the bone marrow and enters a dormant G2 phase, it's basically playing dead. The chemotherapy sweeps through the bloodstream and the marrow, annihilates all the active cells, but completely ignores the sleeper cells waiting in G2 arrest.
It serves as a literal cloak of invincibility. They stick to the walls, pull the cloak over their heads, and just wait for the toxicity to clear. And there is an equally insidious molecular layer operating inside the cell while it hides.
The gene connection.
Right. The paper details how high levels of CD81 correlate strongly with elevated expression of a gene called LAPTM4B, and together they activate the STAT3 signaling pathway.
The STAT3 pathway is a notorious driver of treatment failure. It operates as a transcription factor, diving into the nucleus to turn on genes that promote stemness, basically locking the cell into that primitive root-like state we talked about. But even more critically, it upregulates the cell's efflux pumps.
And those efflux pumps are essentially the cell's internal bouncers. Even if a chemotherapy drug like daunorubicin manages to breach the cell membrane, the STAT3 pathway ensures the cell can physically spit the toxin right back out before it damages the DNA.
That is wild.
It is. So CD81 provides an external anchor to hide the cell while simultaneously wiring an internal defense network to pump out any poison that actually gets through.
Which brings us back to the ultimate LSC problem. If CD81 is the Velcro protecting these LSC roots, what happens if we target it? Can we chemically rip that Velcro off?
Well, to do that safely, we circle back to the therapeutic window we discussed earlier. The researchers mapped the surface profile of the leukemia stem cells. They isolated them by looking for cells that are CD34 positive but lack mature markers. So they are CD38 negative, CD90 negative, and CD123 positive.
Okay, got it.
This specific signature isolates the most primitive, undifferentiated roots of the leukemia. And they found CD81 is highly enriched on these exact cells. But the crucial breakthrough is that CD81 is essentially absent on healthy, normal hematopoietic stem cells.
Okay, but I have to play the skeptic here. We've seen miracle targets in mice before that end up causing catastrophic toxicity in humans because they wreck normal, healthy cells. How do we know this won't do the same? I mean, immunotherapy in the bone marrow is historically a minefield for toxicity.
It's a very valid concern. But what's fascinating here is how they rigorously tested the safety of their proposed immunotherapy solution. They used an anti-human CD81 monoclonal antibody,
Which is an engineered immune protein designed to circulate, seek out, and bind exclusively to the CD81 target.
right? And to address the toxicity concern, they exposed healthy human bone marrow directly to this antibody and ran colony-forming unit assays.
And for you listening, the colony-forming unit assay is a brilliant way to test functional viability, not just whether a cell is alive or dead. You place the stem cells in a semi-solid gel matrix and wait to see if they can still divide, differentiate, and form thriving colonies of diverse blood cells over a period of weeks.
And the assay results showed zero toxicity. Zero. The normal human stem cells exposed to the anti-CD81 antibody formed colonies perfectly. Their cell cycles were not interrupted, and their ability to generate blood was completely unhindered.
Yeah.
Because they lack the CD81 marker, the antibodies simply bypass them.
Here's where it gets really interesting, because ensuring the treatment is safe is only half the battle. It actually has to eradicate the leukemia.
Right, safety doesn't equal efficacy.
Exactly. So for the grand finale experiment, the researchers took mice heavily engrafted with human AML. They administered a brutal regimen of standard chemotherapy cytarabine, doxorubicin, and a targeted drug called gemtuzumab ozogamicin. But for the experimental group, they added the anti-CD81 antibody into the mix.
They attacked the disease architecture from two entirely different angles. They used the chemotherapy as the primary toxic payload, but they used the antibody to strip away the survival mechanisms.
So the antibody bonded to the CD81 on the leukemia cells, physically blocking those Filipodia-like Velcro hooks from latching onto the bone marrow fibroblasts.
Exactly. Without that structural anchor, the leukemia cells couldn't trigger their G2 cell cycle arrest. They were forced out of their dormant sleeper cell state and remained active, dividing cells.
They were locked out of their bunkers right out in the open, precisely when the chemotherapy swept through.
The synergistic effect was undeniable. The combination therapy completely dismantled the cancer's defenses. It led to a massive reduction in tumor burden, it significantly delayed the onset of relapse, and it vastly improved overall survival rates in the mice. And crucially, they validated this using patient-derived xenograft models.
Oh, the PDXs, meaning they weren't just relying on lab-grown, immortalized cell lines like cell line-derived xenografts or CDXs.
Right. They took primary leukemia cells directly from human patients and grafted them into the mice, and the treatment still worked. The antibodies successfully disrupted the real-world biology of the human disease.
It is a remarkable shift in how we approach overcoming drug resistance. So, just to summarize the big takeaway for you from this deep dive, we really need a fundamental reframing of what CD81 actually is. It is not a passive biomarker on a lab report predicting a grim outcome.
No.
It is a highly active physical shield. It provides the microscopic Velcro that leukemia stem cells require to tether themselves to the bone marrow, arrest their own cell cycles, and basically hide from the storm of chemotherapy.
And by targeting it with specific monoclonal antibodies, we might finally have a way to pull those LSC roots out of the garden for good without harming the soil.
Exactly. But before we wrap up, I know you had a final provocative thought you wanted to share, building on this source material.
Yeah, I do. Because beyond the immediate therapeutic potential for AML, This paper leaves us with a really profound biological concept to mull over. We mentioned early on that CD81 belongs to a family of proteins called tetraspanins.
Right.
Well, the defining feature of a tetraspanin is that it weaves back and forth through the cell membrane 4 times, acting as a critical structural protein.
So they aren't just floating around randomly, they actively shape the surface of the cell.
Precisely. Tetraspanins are known for creating something called tetraspanin-enriched microdomains. You can think of them as molecular event planners. They act as a physical scaffold on the surface of the cell membrane, pulling together various integrins, receptors, and signaling molecules into tightly organized, highly functional clusters.
Oh, wow. So CD81 isn't acting alone. It is building a specialized microscopic hub on the surface of these leukemia cells to coordinate all these survival tactics. The adhesion to the bone marrow, the STAT3 pathway activation, the efflux pumps.
Which raises an incredibly exciting question for the future of oncology. If CD81 is the primary scaffold holding this entire survival network together, what other hidden, dangerous proteins is it pulling into these microdomains that we haven't even cataloged yet?
That's a huge question.
Right. We know about LAPTM4B and the fibronectin binding, but the architecture of a tetraspanin microdomain is vast and complex. Could dismantling CD81 be the key to unmasking entirely new vulnerabilities in cancer?
Because if you take away the foundational scaffold, the entire building collapses. It turns out if you want to permanently defeat a complex mutating cancer, you don't necessarily just invent a more toxic poison. You find a way to map its survival networks, locate the structural hubs, and rip off its cloak of invincibility.